Thank you very much. Indeed, a pleasure to be here. My name is Mattias Lundin, and I'm the CEO of Lumito. Lumito is a medtech company here from Lund. We are 15 dedicated, hardworking people that have developed a new revolutionary, unique digital imaging for tissue diagnostics and tissue analysis. And we are quite close now to launch our very first product. So we will offer soon a more precise and more granular image of tissues than what is used today. And we will launch this during next half year in 2023 towards the research community. So what is the product? Yeah, the product is an image on a screen, the way we are looking at it here. And how do we get to that image? Yeah, well, there are two parts that are very crucial and essential here. It's a scanner and it's nanoparticle reagent kits that are applied on the tissue that you would like to look at. So on the left here is the laser or the scanner, and the scanner is two things. It's a more traditional LED-based bright field scanner, and it's also a special laser, near-infrared laser within the scanner. And then we are having down here the reagent kits. So it's a reagent including nanoparticles that are applied on the sample that you would like to look at. So over here we are having the slides, the glass slides that you are having your tissue on, and then the researcher or the pathologist select a biomarker that they would like to look at, and then our reagents with a nanoparticle are then binding to that that conjugation to that biomarker that the end user would like to look at, and then combining them the laser with the nanoparticle. Then you're getting the trick, if you will, and so-called anti-Stokes shift, which is making the particles to glow, if you will, and then the possibility for us to take images on that. So the specific area that you would like to look at is getting very, very clear and bright and granular. There are quite some advantages with the technology. Here we are having 4 distinct product advantages. So we are providing higher level of contrast and granularity compared to traditional immunohistochemistry. So it's a good, I mean, it's getting very, very good visual images, but it's also a very good base for quantification and making AI algorithms based on that input. And the images are only showing what's intended to be shown. So what you see on the green here, so is then the nanoparticle. UCMP stands for up-converted nanoparticle. So that's the up conversion is the laser and the nanoparticle effect. And it's only showing the area that, or the granularity on the biomarkers that you would like to look at. The rest is getting black, so it doesn't show at all. And then we are able to combine both the more traditional way, hematoxylin staining on tissue, and then our way of doing it, and then combining, so you are able to see morphology and where you are on the tissue, and then toggle on and off, looking at only particles and the entire thing, and in a combination. So we are providing more accurate and relevant information in the very same slide. This is enhancing the efficiency, so less slides and getting more effective. Two modalities I've already spoken about. And the good thing here is that it's also within the traditional and regular workflow within the labs in research and also in clinical settings. So it's the same kind of methodology, that the way you are staining your slides before they are under a manual microscope, which is the traditional way, which is more or less 90% of the market today anyway, or in the future, and in what's cooking and going on today, making it digital. So in a digital scanner that we are also having and providing. When it comes to business models, so we are having a scanner and a reagent kit. And we will offer both capital sales and then recurring revenue, because they need the reagent kit for every slide. It needs to be applied a liquid, right? The other alternative that we will offer is to have a reagent rental. So you are placing the instrument at the laboratory, and you're setting up a system that they are financing the instrument via the reagent kits. So the consumption, and then of course a higher pricing on the reagents. So those are the two models that we will offer the market. A little bit also about the strategy that we have, I would like to share with you today. Here we are having five pillars. We would like to offer our solution to the entire market. And the entire market is close to 40,000 labs globally, both research labs and clinical labs. And it's a huge market with some $5 billion annually, with a nice growth of 6.6%. We are, as a sales model, we are going with direct sales in Sweden and Scandinavia, whilst going outside the Nordic countries, we are setting up distribution and searching for distributors that are having larger market shares in order to having access to a larger market. The third area is patents, IP. So we are actively working in coming up with patents when it comes to our technology. Today we are having 3 patent families, but would like to extend that, of course, and working actively on that. The fourth area is innovation. I mean, the whole thing is coming out of an innovation, and we would like to pursue the same journey going forward. And the areas that we are innovating around is, of course, in the core technology, which is then the nanoparticle area, to explore that further, to add more particles, to extend it to multiplexing, and so on. And the other area is image analysis. So having the good foundation that we provide with our technology is a good base for more quantification and AI-driven technologies. And that's an area that we are also pursuing. When it comes then to everything that is not our core technology, we are outsourcing that. So production, for example, we are having a reagent producer in Finland and a scanner producer in Sweden that we are working on. And then again, distribution, sales and service activities will then be also outsourced to the ones that are having good resources to doing so globally. This is the market that we have selected as our first market. So if you recall the 5 billion US, then 26% of that is the European market, which is some 1.3 billion US. And then a quarter of that is the market that we've been selecting as our first market. And that's what you're having in front of you here. That this is the research market in Europe. That's the market that we in the first priority are entering into. The largest share is pharma companies, CROs, and then academia and so on. It's a nice market and having a growth on 5%. In September, we exhibited our technology, our solution, for the first time outside our own office, if you will. And this was in Basel, in Switzerland. And I have to say, we got a lot of interest, both and curiosity, both from potential end customers as researchers and pathologists, but also from the industry. We had market leaders coming in to our booth, our small booth in the corner in this huge congress place in Basel. And so say, we heard about that you have something new. I said, yes, we do. And then you started to, and you could see in their faces, they were quite impressed and interested. I even got a quote from one of them saying that, once again, innovation comes from Sweden. I think that was really an acknowledgement. Are we losing time here? Next one, we're going in next week. We're going to London to the digital pathology. What are we focusing on right now? Sales in Scandinavia, maintaining the contacts with the leading partners that potentially could be distributors, but also developing partners, if you will. And then present results from our pre-studies, of course. And then the fourth, final, and then not the least, of course, is manufacture, to be done with the products, CE mark, and launch. A little bit about the pre-studies. Here is one of them. I quote Daniel Lund from Umeå University. Lumito's technique has provided better possibilities compared to other immunohistochemical methods. Our technology was the best they ever tested, and this will be presented, a publication soon in Cancer Immunology Research. Here are the three others that we are working on. Renal biopsy, the first international one, University Hospital Coventry, Warwickshire. And then we are working with the Dalarna, combining standard methods with our technology on breast cancer. And then also with Uppsala University when it comes to PD-L1 and lung cancer. Pathlake, it's a digital pathology excellence center, I would say. And key leader here is David Sneed, which is a key, really a global key opinion leader for digital pathology. And he encouraged us to apply to be members in Pathlake. And after quite a process of presentations and discussions, and finally we got the membership and got approved. And that was a feather in the cap for us, for sure. Ladies and gentlemen, this was my last slide.
Thank you so much.
And thank you very much for listening. And I hope, finally, that I got your attention, and that you also see the potential and the possibility that we have in the market. So thanks. And I guess this is questions now.
So I will start us off then. I understand that you delayed the launch a little bit.
Yes.
Would you like to explain this delay?
Yes, it's really about I would say three things. When coming up with a final prototype, with a final design after verification. We wanted to make some final small adjustments on the design in order to optimize the scanner, and then getting parts and getting those parts done in order to put them again on the that is you're getting delays in delivery times. So I think that's one part. The other part is then when you then delaying for a few weeks, then you are missing your slots when it comes to these the test houses that are doing all the tests when it comes to EMC, electrical safety, etc., etc. So I think it's actually those two things that finally ended up in that this is not realistic. We will not be able to do this before Christmas, and therefore we need to take a few breaths and postpone it a little bit. Yeah.
I see. And now you're launching in both Europe and Scandinavia at the same time?
Yes.
Yes.
Now we are starting here.
Starting here.
Yeah. So hopefully across the street.
Okay.
Yeah. Let's start there. And then we go further.
So we have a question over there.
Yes.
Yes, hello. My name is Tatiana Kovtuch. I am clinical manager of Neola Medical, and I have one question. How do you think your technology allow to use it in developing countries? I mean, maybe outside of Europe.
How we will be allowed, or
I mean, financial side of your technology.
Yeah. Yeah. Well, you know, I think when it comes to the way we are setting up the business model, I think over time it will also be possible to offer it to developing countries. But our first priority is Europe and then US. I would say. But for sure, like I said earlier, we believe that we would like to offer this for the entire market, and we'll take that step by step. Yeah. Thanks.
We have another question over here.
Oh, I actually have two questions. Hope that's okay. So first question, how does this UCMP compare to currently available IR dyes like the Alexa Flores, qubits, etc.? Second question is, have you looked at this in potential in vivo imaging applications? Do you have any advantages in, say, like depth penetration into tissue and all that?
Yeah. First question, I would say the advantage is handling. It's one area. It's quite complicated and messy to handle IF. The other part is that you usually have challenges when it comes to autofluorescence. So it's also giving light from the background of the tissue. So we are just getting on that specific spot, the nanoparticle spot, and the rest will be completely nothing. So I think those two things are the advantage there. In vivo, I mean, of course, I mean, way back, it's been on the table. We selected to go the path that we've been taking so far, but I wouldn't say it's not unlikely that your question is valid. But we are not working on that currently.
Okay,
yeah.
Thank you.
Thank you.
Okay, so we have one last question over there.
Yeah, it's just a small technical question as well.
Okay.
About a lot now is about multiplexing. So how is your technology working in multiplexing different types of markers?
Yeah. Today, our technology is having one marker, and then you can combine that with other IHC. But our technology provides one today that we are launching soon. But we are also testing, other particles, and so it's definitely doable to add more particles and having more multiplexing in that perspective. But the combination is that we're having our indication, and then you combine it with other things. And the good part is that, for example, DAB. Then you are able to when you're applying that, then it covers a lot of the tissue and you don't see. So, but with our technology, you are able to toggle it on and off. But back to your question, it's only the first thing. Yeah.
Okay, Thank you so much for coming.
Thank you very much.
Thank you.
Thank you all.